Prior to beginning the activity, please be sure to review the faculty information and disclosure statements, as well as the learning objectives. After listening to the activity, complete the post-test by clicking the Earn Credit link in the episode description. Downloadable slides and resources are also available. The following presentation is copyrighted by Medscape. No use, broadcast, or recording of this presentation or any part thereof is permitted without the written authorization of Medscape.
This activity includes discussion of therapeutic products that have not been approved by the U.S. Food and Drug Administration, off-label uses of approved products, or data that were presented in abstract form. These data should be considered preliminary until published in a peer-reviewed journal. Readers should verify all information and data before treating patients or employing any therapies described in this or any educational activity. A qualified healthcare professional should be consulted before using any therapeutic product discussed herein.
The following presentation is part of a certified educational activity provided by Medscape Education and supported by funding from the U.S. Food and Drug Administration.
Hello, I'm Dr. Russell Cohen, Professor of Medicine at the Pritzker School of Medicine, co-director of the Digestive Disease Center, clinical director of the Inflammatory Bowel Disease Center, and co-director of the Advanced IBD Fellowship Program here at the University of Chicago in Chicago, Illinois. Welcome to the program entitled Best Practices for Integrating Biosimilars into Clinical Practice and Interprofessional Perspective. Joining me today are two outstanding experts. First, Stephen Feldman. Dr. Feldman is professor of dermatology and professor of pathology and professor of social sciences and health policy and public health sciences at Wake Forest University School of Medicine in Winston-Salem, North Carolina. In addition, we have Dr. Leonard Calabrese. Leonard is a professor of medicine at the Lerner College of Medicine at the Cleveland Clinic. He's the director of the Cleveland Clinic's section of clinical immunology in Cleveland, Ohio. Welcome, gentlemen.
Good to be here. Pleasure.
Terrific. So why don't we start off with Steve? So Steve, this program is about biosimilars, but why don't we just start with just a quick introduction to what is a biologic?
Biologics are these really large, complex, not just proteins, but glycoproteins that involve tertiary folding and quaternary aggregates. They're very complex molecules. They're not like small molecules like aspirin that you can duplicate. These molecules are so big and so complicated that nobody can duplicate them.
Sure. So that was a biologic. So then what is a biosimilar?
Yeah, a biosimilar is not the same as the originator. It's different in a way because these biologics are so complicated, they can't be duplicated, but the biosimilars are very similar. When I first started hearing from other dermatologists about this, they didn't feel comfortable with biosimilars because they weren't the same as the innovator. But once you understand that nobody can duplicate them, not even the innovator company, you realize that there's variation in the batch to batch with the originator drug. And the biosimilar is basically so similar. It's almost like having another batch of the innovator.
So are biosimilars generics?
No, they're not generics because generics would be identical. I think we have to understand that the innovator product shouldn't even be called the innovator because it varies from batch to batch. In fact, even within a batch, there's variation in the glycosylation. If you run it through an HPLC column, here we have a Tanacept, you see there's multiple different peaks and the peaks vary from batch to batch. The same thing happens with adalimumab. But the biosimilars are so similar. So if you look at different batches of the innovator, you find just slight variations in their binding affinities to their target. And you see that the biosimilar pretty much falls into the same range as the different batches of the innovator product. And then when you look at the pharmacodynamics, you can't even tell there's multiple lines here. The biosimilar stays in the blood, basically the same as the originator for all clinical practical purposes.
Yeah, Steve, I got a question that was sent to me before we started our program from one of my colleagues asking about anti-drug antibodies. So if someone makes the anti-drug antibodies to, let's say, adalimumab, will they make anti-drug antibodies to the biosimilar for adalimumab?
Yes, they will. Yeah, if somebody's drug stops working, the innovator drug stops working because somebody's developed an antibody to it, time to switch to another drug. Interestingly, if you look at the rate at which there's antibody against the innovator versus the biosimilars, you actually tend to see less antibodies against the biosimilar than you do against the innovator drug.
So, but just to be clear, if I'm a clinician and I'm using a originator drug and my patient makes neutralizing antibodies, the answer would not be to switch to a biosimilar or vice versa.
Exactly. That's exactly right.
Okay, that's a common question that we get, and I'm glad that you helped clarify that for our audience. So, what is this slide about drug survival? What does that mean?
Yeah, drug survival is how well people stay on the drug over time. And this is something that's measurable in large claims databases and in registries. And you can get a sense of how well the drug is performing because if the drug stopped working uh, due to poor efficacy, due to anti-drug antibody, or if the patient had to stop the drug because some side effect, you would detect that in the claims, then the drug's no longer surviving in the patient. So I think drug survival is a pretty good measure that the drug is still working, it's still effective, and it's not causing any side effects. And you could see that the biosimilar performs just about the same, maybe a tad better than the innovator drug.
So how about a little summary about the background you provided us?
Yeah, there's several take-home messages here, and the most important one is that the biologics, the innovator drugs, are so large and so complicated that nobody can duplicate them, not even the innovator company. So that the biosimilars are kind of like another batch of the innovator, only we have a tremendous amount of data showing that the biosimilar is similar to the innovator drug and performs similarly in clinical trials and in test tubes.
So, Steve, you're a dermatologist, correct?
Yes, sir.
Right. And one thing you may notice is that a lot of these biosimilars were tested in our inflammatory world in dermatology, such as psoriasis, or in rheumatology for rheumatoid arthritis. The endpoints are easier to see. The expected change in disease state can be over a 3 to 6 month period of time. But I'm a gastroenterologist. I treat Crohn's disease and ulcerative colitis. So, what is the mechanism by which these drugs, which get approved for rheumatoid arthritis or psoriasis, can then have their data moved over to Crohn's and colitis?
Yeah, it's the drugs really should be tested in psoriasis if that's one of the indications, because you want to use the indication that will be most sensitive for detecting a difference between the innovator and the biosimilar, if such a difference exists. And because you can look at the psoriasis and see it, it's probably going to be a more sensitive indicator than asking people about their pain and their rheumatoid arthritis or about their diarrhea rates. Um, but what's I think most interesting is that when people are worried about, well, they're testing it, not, I want it tested in my disease. I don't want it just tested in Steve's psoriasis patients. Remember, the innovator product varies from batch to batch, and you have no study, more likely than not, on the current batch of the innovator drug. And yet we extrapolate for that batch all the indications of the innovator. With a biosimilar, you're actually getting a clinical trial done in the most sensitive disease typically to prove beyond any doubt that the drug's going to perform similarly.
But the determinant that you use is extrapolation. That's what the FDA had given the biosimilar manufacturers as guidance, saying that while they could be tested in, let's say, psoriasis or rheumatoid arthritis, in order for them to extrapolate their results, they did have to provide some type of data suggesting that it should be comparable in these other diseases, because that's one of the concerns let's say, the gastroenterologists have, because none of the biosimilars are initially tested for their approval through the FDA or the EMA in gastroenterology. There's a similar issue that we see biosimilar manufacturers are sending us advertisements that their agent is interchangeable. What does that mean?
Yeah, interchangeable is a regulatory definition where the FDA has recommended that some additional studies be done where you switch between the innovator and the biosimilar back and forth and show that there's no change in the clinical activity. And I believe again, if you understand that even the innovator can't be duplicated from batch to batch, we never concerned about changing from one batch of the innovator to the other or changing back. So this idea that we should need to do that for the biosimilar seems a little odd to me, given that we have more data on the similarity of the biosimilars than we do for the batch to batch variation within an innovator product.
Right. So I think for the purpose of the audience, and if you're in the United States, currently interchangeability laws do exist, and pharmacy laws are actually state-based. So the state actually regulates notification about switching to a biosimilar, and if and when does the physician need to be notified, and does the patient be notified. The principles of interchangeability are that they don't have to necessarily notify you to give a different order. But let's move over to Leonard because Steve, you mentioned these multiple switches. Leonard, what can you tell us about these data?
Well, just to add a little bit to this conversation, these biosimilars have been with us now for a good number of years. And I can recall that, at least in our community of rheumatology, immunology, there was a great concern about extrapolation, taking a TNF inhibitor that's been shown in psoriasis and then using it in uveitis or a totally different biologic paradigm of disease. And interchangeability was a great concern. And from my perspective, clinical perspective, I have not seen any biosimilar that has been approved on the basis of robust clinical data and pharmacokinetic and pharmacodynamic data that has failed those initial tests. So I was very skeptical at first, and I've been more comforted by this. The other uh, uh, area that would be of concern from a pharmacologic perspective is multiple switches, because this happens now. The introduction of a biosimilar is by and large out of the clinician's hands and into the hands of third party payers and the big pharma who are creating these pipelines. So it is not uncommon for me to see a patient of mine who was on an originator drug and then was segued to a biosimilar and yet to be segued to another biosimilar. And, those studies actually, there are very few examinations at a pharmacokinetic level and immunogenicity level, but clinical experience has demonstrated that there doesn't appear to be a palpable um, change in efficacy. And we should be aware of that because patients will question us of what is this drug I'm on? This wasn't the drug that I was just on 2 months ago and I was not the drug I was on a year ago. So far, so good on these data. What's your perspective?
Well, so the multiple switches in the studies were only done between an originator and a single biosimilar, back and forth between biosimilar and the originator. And those graphs, I think, are shown here nicely on the slide. What we don't really have is what you refer to, which is now what the payers are doing, is having the patients hop, skip, and jump from different biosimilar to different biosimilar. Um, in our world, ustekinumab biosimilars now, we might have a patient switching every 2 to 3 months to a different biosimilar just because the contracting. I think that's something that we can talk. As we move forward in this discussion, we can talk more about the real world of biosimilars now that they're here.
Yeah, I don't worry about those multiple switches so much from the biology standpoint, because if you're binding the target the same and you're staying in the blood the same, yeah, it's going to work the same. And we patients have been switching from one lot of their originator to the next lot, to the next lot, to the next lot. We didn't pay that any mind, even though there's lot to lot variation there too.
Yeah, yeah. So it's interesting because I think all of us have mentioned, well, the patient feels and the provider feels, but Leonard, when you have a patient coming in for your treatment for whatever specialty it might be, when should you consider a biosimilar for a particular patient?
Well, Russ, in the reason that we have biosimilars is to lower overall health costs. And the Europeans and single state payers have done this mightily. And because these drugs are less expensive at the from the producer to the pharmacy, there are savings. On the other hand, these savings, by and large, are not in the hands of our patients. And I haven't really had any patients who told me that being on this different drug is cost savings to them. And I don't hide my agreement with them. Yet, on the other hand, we had been working in a healthcare system where we select a drug and then the payer decides whether this is the drug that they're going to get or not. And I recognize that anytime I write a branded biologic for which there are biosimilars available, it is very likely that this patient's going to get a biosimilar. So upfront, I will engage with them and frame what is going to happen. It's terrible to have a surprise, and that's where I started.
Well, do you find are there any medical situations where you would say I would require that they get a branded originator or I require they get a biosimilar?
Uh, Up front as the first starting point, no, I would not.
I agree with you. I don't think there's really any legitimate reason to say that, well, this particular patient, they have to get a branded or they have to get the biosimilar either way, because we really have extensive medical data showing that the drugs perform equivalently in so many studies for the approved biosimilars. Just be aware that many products that were proposed as biosimilars failed in their approval and are not on the market. So only ones that made the cut are here. So either Leonard or Steve, do you find any reason to prescribe a originator drug versus a biosimilar as far as medical reasons?
I don't prescribe it for medical reasons. I provide it because the originator has the longest track record and I have the most experience with it. I'll wait for the originator drug and wait to see what the patient's payer will provide.
The originator sort of has the longest track record, but the current batch of the innovator product doesn't have a very long track record at all.
I agree with you 100%, but the database of patients years on the originator is the largest. And unless you have a drug that you can tell me is palpably better, I generally use that as a guiding point.
Yeah, but since every batch of the innovator is different, we don't have years of experience with the current batch of the innovator. So what I would say is if there's a situation where you would say, listen, I want this patient to be on the original batch of the innovator that was tested in those original studies, okay, then I think you'd have a medical reason. But since I can't imagine that situation, then no, I'm just equally happy between the current batch of the innovator and the biosimilar.
Steve, do you prescribe biosimilars?
I prescribe, for example, adalimumab, ustekinumab, and they're available as biosimilars. And whichever one the insurer says they want to pay for is, as Russ was saying,
We're on the same page and the same operating. Russ, do you prescribe? Do you write for a biosimilar?
Because there's so many different biosimilars for adalimumab in my world and infliximab, not as many, and now ustekinumab. Essentially, I prescribe the medication and say to our pharmacists, we have our own specialty pharmacy, whatever one their insurance will pay for. But what type of do you guys had some challenges when you're trying to integrate biosimilars, either from the patients or they're surprised when they're switched and no one told them? There was a comment about the cost of the medicine. Any particular challenges there?
Yeah, I think that there are challenges, and I think that they're manageable challenges. The reality is that in our country, United States, for drugs where there are biosimilars available, third-party payers will control what biosimilar will be given to them. I don't want my patient to be surprised. I want them to know that I am prescribing a class of drugs, and I'm prescribing a drug that has strong rationale for their disease, and this is the benefits, and these are the risks as I view them. And I want them to be aware that it is very likely that you will not get this branded product, but you will get another product, and I will go in to explain it, and I will be positively framing the biosimilar in every way that I can. And things that I don't say is that you're going to get another drug because it's cheaper. Um, and while that may be the driving force, that in itself is negative framing. And early on, patients who were switched to biosimilars who didn't have a robust conversation and believed that they were getting some type of second class drug or a drug that didn't meet all the things that both of you guys have described in terms of defining its equivalence, created patterns of nocebo responses. Yet at the same time, it's been demonstrated clinically and experimentally that if the framing is positive, these drugs are absolutely equivalent. My experience with them has been identical. I don't want you to have concerns over the name of the drug that you get because I'm not sure which one that you'll get. And these are all things that both we can do and our APs and the nurses in the infusion suites. If someone asks hey, the last time I got this biosimilar, what's up with this one? People need to be able to respond to that empathically and with a lot of positivity. So nocebo responses are kept at a minimum.
One of the problems that we've had is the patient experience has tremendously decreased over the past year to two with biosimilars. In that there are some biosimilars, while the originator may be citrate free, their insurance requires them to go on a citrate containing biosimilar, which really is unacceptable. There are in the adalimumab, for example, there's an 80 milligram in a single vial now. Not all of them come in 80 milligrams. So now the patient's going back to taking two shots. Some of the biosimilars don't have the starter kit. So unlike in other inflammatory diseases, in Crohn's disease and ulcerative colitis, the adalimumab loading dose is 160 milligrams. So now the patient's going back to doing 4 pens. Um, the other problem with that we've had is that the patient assistance has either been in some of the biosimilar companies non-existent. Some of them don't allow the copay cards to be applied to anyone under 18, or they only allow the copay cards for their own unbranded biosimilar. So, this is not something that we as physicians expected was going to happen. That the patient experience was going to be undermined. We were told it was going to be the same, and that's actually one of the reasons that I think some of us are having pause with the biosimilars now because of all the shenanigans that's been going on, kind of behind our back. Have you guys experienced this?
Yes, I agree with that 100%. And it's also very distressing when a patient is sent another drug. The pen is different, different, way to administer it. And you've mentioned about citrate, but several of the products are not latex free, and this is something that's not even discussed. That it has to be appraised, particularly if the patient has latex hypersensitivity. And so I think that we have to be engaged and educate these our patients in a positive way.
So, do you guys have teams that work together? Like, how does your interprofessional team work together to help deliver the biosimilars or the biologics to in your clinical practice?
Most of our patients are either outpatients. The infusion group is very facile with this. We have our own pharmacologist that tends to our immunologic patients. Our APs are all over this. And in fact, one of them keeps a flow sheet. It contains everything from citrate to latex to concentrations. And it's posted. So I mean, this is part of the vernacular of what's going on in a a large, busy, immune mediated inflammatory disease unit, just like you guys have. It has to be.
Steve, do you have those resources?
I've got some great resources, but Len's practice is clearly far more proactive in this area than ours is. I practice in a major medical center. It has its own specialty pharmacy, as you were saying, and so we got a lot of support from them for getting patients on drug. But I don't think we've paid much attention to whether they get a biologic, an innovator, or a biosimilar. And so occasionally I'll get a cell phone call from a patient or a text saying, you know, they sent me it's a different thing, and I'm like, "Oh, it's just another name for basically the same stuff."
Yeah, right. I think so too. So, I think it's important whether you're in a small practice, a university practice, or you have Leonard's mecca of inflammatory diseases, to have your team work together with the patient. So that way you can build confidence in biologics and in biosimilars. I mean, clearly, the biosimilars offer a tremendous savings to the healthcare profession. And in reality, the data for the clinical efficacy is nearly identical and numerically, sometimes, as we say, better than the originator drug because the originator is not the one that was made 25 years ago. Nevertheless, I think that it's helpful to be very clear when you're starting a patient on a biologic to say to them, this is the name of the drug. It's off patent now, so you may get a biosimilar. It doesn't matter. Whatever you get, we go with it. That's an easier decision. Switching people, from one to the other, you can get what, as Leonard mentioned, the nocebo effect, where patients presume that because they're getting something different, it's inferior and not working. I think you have to work through that with them too, and say that there are hundreds of patients and now thousands that have had it, and the chances of there being a difference are really no different than the chance of you staying on the same product and having some drift away from your original success as well too. I purposely threw the word generics in earlier. That's taboo. They're not generics. They are highly complex with compounds. And I think that it's important for people to know that not every Tom, Dick, and Harry makes a biosimilar gets it approved. They all undergo extensive testing for efficacy and safety. So just as we're closing up, I will start maybe back with Leonard. Leonard, any closing comments that you might have about this topic for our audience?
No, I feel that anybody that uses biologics should have a good, basic fund of declarative knowledge, as you guys have so nicely, gone through regarding the hurdles that these agents have overcome to be designated as biosimilars. Secondly, I think that for those of us who have patients on biosimilars, that we have to mentally have that in our head that we want to avoid nocebo effects. We want to avoid being pejorative about these drugs or considering them, cheap alternatives to what I wanted to give you. It would be horrible. And talk about them as desirable drugs that have met a good clinical and scientific rigor. There's a lot of resources available for um, clinicians that want to get up to speed on this if they aren't already. Most of us that have been using biologics a long time have done this through the school of hard knocks of having many, many patients on these drugs. So I think we're getting a lot better in that area.
Perfect. And Steve, any closing comments for our audience?
Yeah, I know it's hard to believe, but patients on self-administered medications aren't generally fully adherent. And I think the variation in patients' adherence behaviors and how they handle the drug, the temperature of their refrigerators, there's so much variability there that it totally swamps whatever little variability there is between the different batches of the innovator or between the innovator and the biosimilars. And the main thing is that I hope for all of your sakes that the insurance hassles from where the insurer makes us switch from one biosimilar to another one this week versus next week just isn't too overwhelming for us.
Well, so on that note, I'd like to thank you all for participating in this activity. I'm Dr. Russell Cohen from the University of Chicago. We have Dr. Stephen Feldman and Dr. Leonard Calabrese. I hope you found that this interchange was entertaining and educational, and we look forward to seeing you at further Medscape programs. Thanks so much.